Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. My goal here is to find exceptional people in their fields and interview them and ask them good questions and learn things from them and bring that to you, the audience, that maybe they haven't been able to talk about in just a generic article online, that kind of thing. So today I have Dario Valenzano. He leads a research group at the Max Planck Institute for Biology of Aging and at the CCAD in Cologne, Germany. It's CECAD. He studied neuroscience at the Scuola Normale Superiore in Italy. He did a postdoc at Stanford University in the USA. And the research we'll talk about is he's trying to understand how species in nature evolved long or short lifespans and the role of gut bacteria and how that changes during aging. So we'll go into that. And Dario, thanks for coming.
Yeah, thank you so much for having me.
So what are you trying to figure out about aging? Why certain creatures have the lifespan they do, or is it more of what's happening to their gut bacteria as they age?
Yeah, it's true. I mean, there are different questions there, and one thing that really fascinates me, and I see this type of activity, this research activity that we conduct, really curiosity driven. It's really trying to understand how come different species live that differently. For example, there are species that live just a few hours, just a few days. Some invertebrates, some worms, some flies, mayflies, for instance. And there are organisms that live thousands of years, as far as we know. Some, for example, bristlecone pines in California. There are organisms that can preserve themselves for very long. Some yeast have been seen to survive for hundreds of thousands of years, encased in a special substrate. So, in other words, we try to understand in my group how come these different species evolve these different strategies to survive in different environments, and not just out of curiosity, but also we want to understand the mechanism, the genetic, the molecular basis that underlies this difference. So this is one thing, and so on one end, the genetics basis for these differences in lifespan across species, but then on the other hand, we also want to know how can we manipulate aging and lifespan in species like our own, for instance. How can we come up with new strategies, new methods to manipulate lifespan and aging, and therefore reduce the risk for many diseases?
Well, so what's been noticed when you look at the extremes, creatures that live only for a few hours or days versus ones that live, for 50, 100 or more years? Like what are there different regimes? Short-lived creatures tend to have these processes and middle ones and long-lived ones. What do you see?
Right. For instance, species that live very short normally they just survive what it takes them to reproduce, right? So at the end of the day, what you need to do is to keep replicating yourself, your genes, and so perpetuating this line, right? That's what really keeps species going. What we are very much interested in understanding is what goes wrong, right? So what fails? So we are very much into failing modalities, right? Failure modalities in different species. And therefore, when we compare the very long-lived ones to the short-lived ones, what we know as a field, what's been recognized by many researchers over the years, is that, which is quite intuitive, is that long-lived species tend to be extremely good at repairing damage that no matter what will occur. So long-lived and short-lived species, they undergo external, insults, right? So they continuously receive insults from external factors, temperature, light, aggression from other conspecifics, etc., infections, etc., etc. And there is also damage that comes from the inside. So, for instance, when a cell divides, when a cell replicates, or other cellular processes lead to the accumulation of a lot of damage and the risk for continuous damage, right? So the long-lived organisms, the ones that age more slowly also, are the ones who are able to repair themselves better. So they're better suited. They have really complex and sophisticated machines, molecular, cellular, and physiological machineries to repair damage that no matter what will occur to them. So we try to understand, how these differences in repair differ among these different species. So this is one thing that many laboratories, many groups have been investigating. How can we improve DNA repair, protein repair, etc. And then we want to understand. Yeah.
Well, okay. When an organism is, early on in its lifespan, is the DNA repair, for instance, sufficient enough for the organism to do well, or is it only when it gets old? Whatever old means for a given creature. It seems like, for us, that's when our repair systems don't function as well. And, for dogs and for other creatures, it seems like when a creature gets quote unquote old, for that kind of creature is when it has problems.
That's right. So this is one way of looking at it, right? So you can say when you get old, then the repair fails, right? But then what it means really to get old. So probably what happens is that when repair fails, then we start having all that, that whole range, that whole array of phenotypes or traits or characteristics that we recognize as being old or being sick, right? Which is a collection of diseases, if you want, of little failures, right? So aging can be like, the way you were posing your question is aging as a trigger for failure of repair, but there is an intrinsic actually crosstalk between all these repair mechanisms and initiation of the aging process. And to answer your question, what happens early on in life, even if you look at the germline, right? So those specific cells in our bodies, but not just in humans, but in any species in the world, the germline is really good at repairing damage that occurs to them. For example, DNA damage. They are extremely good, extremely protected from insult. And the reason for that is because they have to transmit the genetic information to the next generation, right? So there is a lot more pressure, if you want, selective pressure for the germline to stay clean from these insults, from these mistakes in copying, for example, as opposed to other cell types in the body, like a skin cell or a liver cell.
Well, I guess it makes sense. That's the great hope is, you want to keep the germline as pristine as possible so you can reproduce.
That's right. That's exactly right. And so by trying to understand what mechanisms are there so that this germline can sustain itself for a very long time, we can probably design ways to treat also somatic cells, so the cells of the rest of the body, to keep themselves clean, right? So what happens, for example, during cancer, many types of cancer, is that errors start accumulating, and you have these cascades of events that lead to reprogramming of these cells into new types, into new identities that self-replicate beyond their actually designed capacities, and then they escape the surveillance from the rest of the body, and then damage start to happen. So in my group,
I have a quick question here about the germline. If you look at males and females of a given species, once the male's body, their cells wouldn't know that they've successfully reproduced, but the female's body could, at least in certain animals. Has anyone studied to see if there's a change in preference of the germline cells once, an organism has successfully reproduced, been pregnant and, carried their young to term? I mean, does their body change in such a way where the germline is not as diligently preserved?
Right. So there are organisms indeed that once they reproduce, they actually trigger a whole new process, right? So that leads to oftentimes to demise. For example, many plants, once they bloom, once they actually reproduce, once they are fertilized, they actually die. They let themselves go. This is the case for bamboo forest, for example. They live about 100 years. And then once they reproduce, they actually release factors that kill actively the whole bamboo plants of same age that have reproduced, right? And so you have this recycling of forests every night. Also in animals, this can happen. For example, there is the charismatic case of the Pacific salmons. Once they reproduce, they actually once they home back in their own rivers, they reproduce, and then they undergo this dramatic phenotype of accelerated senescence. This seems to be like a trigger to let themselves go. This is one way to think about it. Another possible way to think about it is that actually they stop repairing themselves, right? I think it's slightly different, but it actually leads to similar outcomes. So there is definitely like a clock that can actually click once you reproduce. This happens in certain species, definitely doesn't happen in all the species. So if you, for example, to address your question, if you prevent a species from, like an individual from reproducing, it wouldn't necessarily live longer or differently from an individual that has, except for salmons and this specific case of the bamboo plant. So if you're programmed to die once you reproduce, then of course preventing reproduction will extend your lifespan. But if you're not programmed to trigger that old process of demise once you reproduce, then even if you prevent the reproduction event, you won't have this catastrophic.
Right. Okay. So if you look at, let's say, people when they're young, as you said, and I'm guessing this is what you said, their bodies are very protective over error accumulation in the sperm and egg cells, let's say. But once, as people age, for instance, or as other organisms age, do they become less protective over damage occurring to their germline cells? And is that a function of aging?
Right. So in humans, for instance, age of both man and woman affects to some extent the germline, the number of errors of DNA mutations that are present in the new gametes, in the new germ cells. So there is also like an accumulation of damage. Although germline is extremely good, extremely protected from damage, later on in life, so older individuals definitely have higher chances. For example, women notoriously have high chance of developing this chromosomal aberration. So these changes in the chromosomes structures, some they fuse, some they split, that can lead to either miscarriages or diseases in the offspring. The older the woman, the higher the risk there is for this type of issues. Similarly, in men also, it's been shown that men at a later age, they actually affect the survivorship of their daughters in particular, and I will explain what I mean by that. So older fathers will actually have slightly shorter-lived daughters than younger fathers, and the reason for that is that they are transmitting to the daughters the X chromosome, which is a large chromosome. This large chromosome will actually have a lot of, just like every other chromosome in that the father is transmitting to the daughters, will have a lot of errors, okay? Just like every other chromosome. However, why daughters and not sons is because the son is just inheriting a small Y chromosome that has very, very limited information. Instead, the daughter is inheriting from the father a large chromosome with a lot of important information, okay? So like an older father that has an X chromosome that has accumulated a lot of errors over time will actually have slightly lower lifespan than a daughter of a younger father. So these were studies that have been published a couple of decades ago.
Where's the advancement and understanding that you're on the trail of in regards to aging? What are you trying to figure out specifically about it?
Well, I mean, the questions we try to ask and to understand are twofold. One is how species evolve different lifespans and aging strategies. So what is the genetic basis and the evolutionary trajectories that lead species to be long-lived or short-lived? And the other question is how can we manipulate lifespan and aging in species experimentally? How can we intervene on improving what we call homeostasis, right? So this process of balance, of equilibrium, of that manages to keep all the system in check. So for the first part, the evolutionary part, we are very much focused on short-lived fish that we study in my lab. It's African turquoise killifish, it's called. It's the shortest-lived vertebrate that we know of. So it's a fish that lived just about 4 months in the laboratory, but also in its natural environment. So we have developed together with other groups this fish as a new model system for aging. And we use it to study how species evolve short lifespan, because this fish is a particular case of evolution of short lifespan invertebrates, which are organisms like ourselves, right? And so what we have found is actually we have studied and understood how this species has become so short-lived. And the long story short, actually, what we found is that Darwinian evolution has not much to do with actually why the species became so short-lived. It seems that the species became short-lived out of an accident. So they live in very particular environments, and there is no selective pressure. There is no advantage in being extremely long-lived in those environments, because those environments where they live, and this is the African savanna, give to this fish just a chance to survive in water for a few months every year, only during the rainy season. So these fish are perfectly adapted to grow as fast as they can during the wet season, to reproduce. And then there is no real strong selection to maintain the body for a long time. So you may wonder what happened to the rest of the year. The rest of the year, when it's dry, there is no water whatsoever in the ponds where these fish live. And the embryos survive in the dry mud, okay? So when we think about the fish, we really think about the adult stage of the fish, the swimming one, right? But normally the embryos, the eggs are encased in dry mud. And so what we found is basically that in these particular environments, there is no selective pressure to survive for a very long time. And what it means is that a lot of mutations that normally affect genes that matter for late life maintenance, to maintain the body for a long time in adulthood, these genes that accumulate continuously mutations will not be purged of these mutations in this specific environment. So there is no selection, there is no counter selection to remove those deleterious mutations. Okay, so it's a little bit like what happens in humans. Mutations that affect very, very old life, for example, dementias, right? Neurodegenerative diseases are like a typical example, right? By the time you've reproduced, by the time you realize that you are sick, you have already reproduced, right? So you've already transmitted to the next generation those genes that actually will lead you to your dementia or to your late life occurring disease. And the same thing for the fish, right? So they have late life detrimental consequences due to the fact that selection does not remove this mutation. So what do we make with this information? We think we understand also a little bit about the human evolution by studying this fish, because we understand that when populations undergo these very strong selective conditions like the fish has, population size decreases dramatically. This is also something that happened in the human species. And selection normally under very small population size is particularly inefficient. So inefficient selection would mean that selection is not particularly good at removing deleterious mutations. So we think that part of the reasons why we age the way we age has to do with the weakened selection in removing these deleterious mutations that stick around.
But what factors would, what factors would cause an organism to live longer or live less long? What constitutes a pressure that would change the organism to live longer or live less?
Right. So you can imagine that if you were this fish, for instance, to go back to this example, if you were to live longer, you could in theory keep on reproducing, right? So you could keep on spreading your genes to the next generations, right? So if you had a pond where this fish lived that lasted for a year instead of 4 months, those individuals would be living longer than the others. They would have the chance to reproduce more, right? And so basically what it means statistically, right, is that in the next generation, you have more offspring from the longer lived ones because they have reproduced more, right? So you can see that would directly translate into a selective advantage. You live long and reproduce more, as long as there is a possibility to keep on reproducing for longer.
But what if you have these fish and you're nice to them, you coddle them, and there's nothing that's pushes them towards death? They've got a nice environment. They get fed every day. You tap on the tank and say hello to them. I mean,
that's right.
There's supposedly no pressure then for them to die at their age. So why wouldn't they all of a sudden start living longer?
Right. So this is exactly what we do in the laboratory. So in the laboratory, we keep the fish with plentiful of water. We don't stress them. We don't create environments that are stressful to them. We feed them every day, twice, and they are in a nice temperature, et cetera, and it would still live those 4 months, right? So they have this constellation of mutations that actually decides how long they're going to be living, okay? But what you're asking probably is like, can we suppress this kind of selection for them not to remove those deleterious mutations, right? So in other words, can we award the longer lived individuals and therefore they would be laying more eggs. So this is actually, this could be done in theory. This is called experimental evolution. One could design an experiment in which you have a large tank, like many large tanks maybe, with enough genetically diverse fish, for instance, to breed them and then to collect embryos of the fish. But you would need a lot of generations, right, to do this, to run this experiment. Actually, we do know that in this particular group of fish, these killifish, most of the fish underwent like shortening of lifespan. There are some instances in which lifespan extended actually, and we want to understand what happens in those fish that actually had this lifespan extension. So we want to understand what happens to them. So this is something that we're actively studying. So, if you long live or short live, actually, we see that has a particular signature in the genome. So we can actually tell whether a fish is long live or short live by looking at particular features of the genome of the fish. So we can now recognize that pretty well, and we want to understand how these features came to be throughout evolution. This is very much under investigation.
What about the microbiome? You said that you were studying microbiomes and how they change as we age. Are you doing that in the fish as well?
Right. So we do that in the fish, in mice, and also we started investigating this also in humans. So the reason why we look at the microbiome is because it's of course has become very accessible experimentally over the past few years. Now we are able to, in the past, to study the microbes, you needed to look at the microscope to culture them in your petri dish and do experiments, right? So, but you needed to culture them in a way to know what microbes are in a given environment. So nowadays we can actually access these microbes by simply sequencing their genome or other aspects of their proteins or their RNAs, right? So we can know what microbes and how many of those are in a given substrate surface by simply sequencing. So what we found is that actually there are very, very rich communities of microbes that are in the gastrointestinal tract of pretty much any organisms that has a gastrointestinal tract, and this is a very well known thing. And so what we found is that there are massive drastic changes in microbiota microbe composition over aging. So humans, fish, other organisms undergo dramatic changes in the microbiota composition over time. And there are particular changes that occur during aging. In particular, what we have observed is that while young individuals have a very diverse microbiota composition, older individuals have a less diverse microbiota composition. So young individuals have different types of bacteria, thousands of different types of bacteria, even fish. Instead, older individuals have a few hundred bacterial types, species, if you will. So, and a few species take over the whole community. This is very similar to what happened during infections, actually. During some specific infections, you have expansion of some bacteria that actually then become pathogenic. They can be very problematic.
How do you think that is? Because there was a loss of roles that the bacteria would take up as systems in an organism get damaged when it's older, or like what would be the reason for this?
The reason for the change in the composition?
Is there just less redundancy, but all the same roles are still fulfilled, or are there loss completely of certain roles by the bacteria?
Right. So we have this hypothesis. So we have the hypothesis that actually what shapes the microbiota composition, what really builds it, it's the immune system. So we believe that the immune system of the host participates actively in deciding who are the friends and who are the enemies, okay? So some of the microbes actually are needed. So we need some of these microbes to give us fundamental molecules like vitamins, vitamin K, B, actually are synthesized by these bacteria. We need them for secondary bile acids. We need them for a lot of different things, for certain aspects of digestion, to digest starches, and et cetera, et cetera, et cetera. And so, so the immune system can really tell who's the friend and who's the enemy somehow. So there is an innate way to recognize bacteria and to actually suppress invaders, but also there are ways to foster friends, right? So to foster, to culture, literally, certain types of bacteria. So what we believe is happening with aging is that the aging immune system, for instance, would lead to a decreased capacity to recognize who's the friend and who's the enemy, or decreased capacity to fight the enemies and to foster the environment so that the friends would actually thrive. So we think that this shift actually is a consequence of aging. The shift in the microbiota composition may be a consequence of aging of the host itself. However, what we also have observed is that microbes per se are able to talk to the immune system. So microbes can themselves manipulate the immune system. In fact, when we transplant microbes from young fish, for instance, to middle-aged fish, we can actually promote health dramatically. So we can extend lifespan. So in other words, we treat middle-aged fish with antibiotics, and we provide this fish with microbes that come from young conspecifics. And these transplants of this complex microbial community actually is extremely beneficial to the host.
What happens in the fish when you do that? What do you notice?
Well, they live longer. They live significantly. So what we have done actually has been to look at survival in the fish, and they live longer. So this was one acute intervention. So at about 9.5 weeks of age of the fish, that corresponds to something like, I would say 45 years, 50 years for a human being. We treat the individuals with antibiotics. So we deplete the gastrointestinal tract of this fish from their own resident bacteria, and we provide them microbes from the intestine of young donors. And they stick with these microbes for a whole day. And we make sure that actually they really undergo recolonization. So we test that over and over. And then once we make sure that the recolonization actually happens, then we put the fish back in the normal tanks, and we just follow their life. And they benefit dramatically by this transplant. And it's not just the antibiotic treatment that is beneficial to them, because we of course have our control groups where we treat the fish with antibiotics, and we give them the same age microbes. Okay, so we compare treatment groups that receive microbes from either young or either the same age, or they don't receive any microbes, right? And then we compare lifespan among those guys. Also, we do a reverse experiment where we transplant microbes from old to young fish. And when we do that, nothing happens. So the young fish, they bounce back to where they're supposed to be at a young age. So they are able to reconstitute their own resident microbes. They are supposed to be there at that age. So that's why we think really that there is a active role and active contribution of features of the immune system to actually shape actively the microbiota composition at a given age. But now, this is still an hypothesis that we are testing. So now we are generating fish that lack this or that aspect of immune system, and then we do the transplant in that context, see whether fish are no more able to actually benefit from the transplant.
Have you sequenced the microbiome of the middle-aged fish that you transplanted the young fish microbes into over time to see if it reverts back to the the older profile, or does it stay with the young profile?
That's right. So we did exactly do that experiment that you're referring to. So we could see that there is a acute shift to the younger state, and then it stays for quite some time. So we tested actually up to 7 weeks post-transfer. That for the fish is a very long time. But eventually it will go back to an aged microbiota. So it won't necessarily stay like that for forever. So we have an acute shift that leads to a long-lasting benefit. But what we don't know, for instance, is that if we do repeatedly this transfer, whether the fish would benefit continuously from this replenishment. So this would be an interesting experiment.
I know this is it's not what you're doing, but I wonder what happens to the food that we eat or creatures eat. Has anyone done an experiment where they fed an animal or a person, like let's say old fish, middle-aged fish versus young fish? I mean, I wonder, again, this is way off, but when you're eating something, when you're digesting it, do you think there's any influence on the age of the thing you're eating? Are you getting some of its bacteria that would benefit you if it's younger or older? Are you somehow utilizing any of the DNA that comprises the food that you're eating, and is that affecting you? Has that ever been looked at?
So are you asking whether we eat a young or old individual and whether this would affect our own microbiota? Is that what you're referring to?
Yeah, has anyone done an
Mhm.
experiment like that where they only feed some kind of creature, old creatures, I don't know, let's say something eats, some
Yeah,
animal eats, rats, and they only feed it old rats versus young rats. Has anyone done
right.
that to see anything?
I'm not sure whether this has been done. I mean, of course, when you cook food, the microbial part is completely filtered out. So you cooked food normally, if you cook it for long, you would deplete of most of the.
No, but most animals eat raw food.
Right, absolutely. Yeah, you're totally right. So the in nature, in nature, there is this very interesting question of whether in nature old individuals stick around for long, right? So whether, but to go back to your question, I don't know whether predators select specific. Actually, as far as I know, predators, like big predators, tend to go after the sick and either the very young preys or the sick and older ones, right? So but if they had to choose, I don't know if they would go for the adults, and whether there is a selection to survive on certain types of microbes. That's an interesting. I don't know whether there is this selective choice. So what I can tell you,
I know it's way off what you're doing, but the thought just occurred to me. I wonder if anyone's experimenting with that.
I don't know. So what I can tell you though is that co-culturing, for instance, could be like rather instead of feeding, right? So it's contact, right? So if you were to keep in contact. So for example, mice, if you raise mice in a lab, normally they are coprophagic. That means that they eat their own poo. And so if you co-house in the same cage mice that are young and old, they would actually share the same microbiota in a way, right? So they would recirculate microbes among them. And so whether that would in a way affect the microbes and other aspects of life of the young of the or the old, as far as I know, has not been explored. There are a little bit of issues.
Oh, really?
Yeah.
They haven't even looked on, old mice housed with young mice because of this,
not some, right.
You would think that it would keep the older mice younger longer, right?
Could be. It could be. So it could very well be. There are not so many people working on microbes and aging. Actually, it's very few labs. So it's a very new field of investigation. So a lot of people studying the microbiota study normally diseases or immune system, or for example, people study cancer in microbiota, people study inflammatory bowel disease and microbiota, specific types of conditions. Aging and the microbiota is a very, very recent field of investigation, as really, very, very young. So these are all open questions. What I can tell you though, because we thought a lot about doing this type of experiment that you're mentioning, is that when you do this transfer, you have to when you do this co-housing experiment, you have to factor in a lot of other aspects. So you have pheromones, so these molecules, volatile molecules that can be sensed and that can affect the behavior and the physiology of the other organism, right? So you have microbes, but you have a lot of other things. You have aggression, you have smell, you have a lot of things. So separating the microbial component from everything else, it's not that easy. And that's why we actually do experimentally, we just do the transfer of the microbe to remove out just that component and be sure that we know what we are adding or what we're removing, and we want to know the consequences of that specific intervention.
When you said that older individuals have less diverse microbiomes, I mean, that's just like, no fault of your own. It's just a very surface level analysis. What is needed to really figure out what the difference is? What would you, the metabolites, the metabolome,
right, exactly.
Transcriptome, what would you need to see?
Sure, right. So when you look at the microbes, you can do it in different ways, right? So you can look at the species count that can come from different sources depending on how you do your sequencing. You can look at what genes actually are on or off, right? So you can look at the transcriptome of the metatranscriptome even, right? So you can look at the RNAs that are present in a given time in a given community of microbes, right? So when we talk about of course, you can look also at the proteome and metabolome, et cetera. Those will all give you additional level of information. And indeed, this is exactly what we are doing right now, also in my laboratory. We have actually longitudinal mice cohorts and fish cohorts where we look at each individual mouse and we actually ask how their microbes are changing over time, but this way in an individualized way, right? So can we predict from those specific aspects, microbes, transcripts, or metabolites from the microbiota, whether we can predict how young, how healthy, and how long-lived that individual will be, right? At the end, we will know that information. We will know how long-lived that individual is, or how much it weighs, and how much it weighs varies over time. So now with multiomics, we can really investigate what metabolite or microbial species or this or that weights more in the prediction, right, of future state. But when we talk about diversity, of course, we can discuss diversity in different contexts. What I was referring to specifically was individual alpha diversity. What this means is that these are how many. So this has to do with richness, right? How many different species of bacteria a young versus an old individual has. And there is this constant decline in species diversity in older individuals and in frail individuals. We don't see exactly the same thing in humans, for instance, with aging. In humans, there is a little bit of a decrease in diversity, but what we see in humans happening in terms of decreased diversity is when you are frail, right? So frail individuals, disease actually is more associated with loss of diversity as opposed to healthy aging, as we may say. There is another type of diversity, which is the inter-individual diversity. This is called beta diversity. So how I am different from you in terms of microbiota, okay? And that measure of diversity increases with age. So any two older individuals that have their own individual and depauperated and poor microbiota composition, they are very distant from one another, right? So it's like they sample the initial diversity that were present in the young in different ways. So each individual is a separate trajectory in microbiota subsampling. If what I say makes sense.
Okay, yeah. Their microbiome has gone down a more and more different path.
That's right.
Well, has anyone looked at blue zones? Sequence the microbiome of blue zone people?
Yeah. So there's been some work done by some colleagues in Italy, for instance. So there are these supercentenarian families or cities in Sardinia. People have looked at that a little bit. So they've seen that there is enrichment in specific type of bacteria, and the trajectories are different from that of the control group, the normal living individuals. However, it's very hard to actually compare the supercentenarians to anybody else, right? So how do you extrapolate their trajectory? So there is no longitudinal information from those individuals, right? So we don't know how they were when they were 80, right? What their microbiota was like when they were 80, so that we can compare their microbiota at that state with a microbiota of a non-centenarian 80-year-old individual. So there is a little bit of missing data still to understand thoroughly this. So I think that there is a need of a longitudinal analysis of that it's still lacking, because then you have.
Is it not useful to look at the microbiomes of centenarians versus a normal population?
Right, but these are still individuals that are extremely old. So what I mean by that is that you need a control group, and the best control group would be the centenarian at 80-year-old, for example, the centenarian at 70, the centenarian at 60, and then we could compare it with the matched 60-year-old, matched 70-year-old, matched 80-year-old who doesn't have the luxury to live over their 100 years, because otherwise you have this very, very peculiar microbial compositions of these individuals, but they don't have a special bacterium, right? It's slightly different from that. So, but as far as I recall from the published literature on supercentenarians, there was nothing particularly remarkable to be observed from these individuals in terms of their own microbiome.
Okay. So what's next? I mean, what are you going to be looking at, and what are you looking to figure out in terms of the microbiome and aging?
Well, for example, what really, different things, of course. So in the fish, we want really to know, for instance, using the fish, we can ask courageous questions, the things that, will take shorter time than in the mice and in the human, for example, and we can work on transgenic fish, for example. And so we want to really understand the crosstalk between immune system and microbiota. We want to try and boost immune function with the microbiota. So we want to restore health in a more precise way than doing like a whole microbiome transfer. We want to understand what aspects of the microbiota are key to improving health in the host. And on the other hand, since I told you before that our hypothesis is that is the immune system that regulates the microbiota composition in the first place, we want to know what aspects of the immune system are failing with time that may be leading to the shift in microbiota composition. And we want to play with those. We want to try to extend the functionality that are lost with aging specifically. So this is one thing that we want to do. Another thing we want to do is actually create sort of predictors of health using the microbiota. So we want to know, I want to understand from aspects intrinsic to the microbiota, whether we can tell whether an individual has a liver problem, like a cognitive impairment, whether a person has a high or low risk for this or that cancer, or whether an individual is at risk for, whether we can tell health from the microbes, right? So there is a lot of information present in the microbes, in the stool, for example, that we cannot draw from blood. Another thing that really interests me is actually more interventional then. So we would like to also understand whether in humans there is any benefit in considering in this case specifically autotransplantation. So whether the same individual would benefit from receiving microbes from the same individual at an earlier age or in a state of better health. This could apply, for instance, after antibiotic treatment to recover or even after conditions. So these are two aspects. And when there is a last aspect that I'm very much passionate about, which is also the evolution of the microbes in the gut, right? So we're very much looking also at how microbes actually are evolving actively in the intestine. So intestine is really like a very rich environment where there is a lot of evolutionary dynamics, and there's a lot of ecology and evolution happening in a time scale of the life of an individual. So we are looking at evolution of microbes and the whole microbiota
Well, very good. Well, Dario, what's the best way for people to find out more about your work?
Well, I guess you can check my lab website, which is valenzano-lab at .age.mpg.de, or you can just Google my name. I guess you would find more about it.
Well, very good, Dario. Thanks for coming, and I appreciate it.
Yeah, thank you so much for having me. It was great talking with you.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.